Everyone, welcome back to Pathomad. Today, let us study about growth factors. A brief introduction regarding growth factors is growth factors are required for cell growth and cell division. What do I mean by this? So, for example, this is a cell. This cell to receive, to grow or to divide, it has to receive a signal from the extracellular space. This signal, it can be in any form, like a growth factor can be a signal, or it can be a hormone, or it can be any neurotransmitter, or any other drugs or chemicals, and these are called ligands. So ligands are nothing but any substance that produces a signal. So this signal, it has to come externally, that is outside the cell, to activate the cell. And the cell, it has an antenna on the surface. On its surface, it has an antenna to receive the signal. This antenna is called receptor. So once the growth, here for example, growth factor is a signal. Growth factor, it binds to this receptor on the surface of the cell. It is called growth factor receptor. This binding leads to transmission of signal inside the cell. The cell receives this signal and it has to transmit to the nucleus. It has to transmit to the nucleus. This signal will be transferred to nucleus where the DNA synthesis occurs, mRNA transcription, and the gene expression. Ultimately gene expression is done. This is how the cell signal receives signal from the outside and transfers to the nucleus where the gene expression is done. So, firstly, growth factors, they stimulate the activity of signaling pathways. This pathway, what I have explained, it stimulates the activity of signaling pathway and genes that augment cell survival, growth, and division. These bind to receptors and influence the expression of genes that promote entry into cell cycle, relieve any blocks on the cell cycle progression, any inhibitors on the cell cycle, these blocks are removed, prevent cell apoptosis, that is cell death, it prevents the cell death, and it increases the synthesis of nucleic acids, lipids, proteins, and carbohydrates, which are required for cell division. So growth factors on a whole, it has to maintain the cell growth function. It has to maintain the number of cells growth. Growth factors also have a role in non-growth activities like migration, differentiation, and synthetic capacity. They regulate cell proliferation at a steady state. That means not in excess or not in this deprived state. It has to be in a steady state or optimal state, not excess, not less deprived state. And whenever required, they have to replace irreversibly damaged cells. Irreversibly damaged cells has to be replaced. For that also you require growth factors so that the cells can divide, proliferate, and reach, and they can replace the damaged cells. If this cell proliferation becomes like hyperactive, when it goes uncontrolled, it leads to malignancy. If at all it is within control, like well balanced, then normal cell balance will be maintained. If at all it turns uncontrolled, increased proliferation of cells, that the cell division runs amok, then it leads to malignancy. So when cell proliferation becomes uncontrolled, it leads to due to dysregulated or altered signaling pathways, the growth factors become continuously active. Growth factors, they become continuously active. So the growth factors can also act as proto-oncogenes. That means like they favor carcinogenesis or tumor formation. The gain of function mutation means hyperactive of this growth factor. It leads to malignancy, so it acts as a precursor to malignancy. So hence they act as oncogenes. So this is a table taken from Robbins. It has 8 factors, explained about 8 factors. First This is epidermal growth factor. Second, transforming growth factor alpha. These both belong to the same family. And these, where are the sources? They are produced from the activated macrophages. Common, I've highlighted the common things. Activated macrophages are in common for this both. And what does this epidermal growth factor do? Epidermal growth factor, it is mitogenic for most of the cell types. Most of the cell types, mitogenic means it increases cell division, mitosis. That means it promotes mitosis. That's why mitogenic for most of the cell types, especially epithelial cell migration, stimulate epithelial cell migration, and also require for wound healing, granulation tissue. Transforming growth factor alpha, again same macrophages is a common thing. They stimulate proliferation of hepatocyte. They stimulate, remember, proliferation. Only proliferation of hepatocyte they stimulate. That is the start. This is third factor is hepatocyte growth factor, also called scatter factor. Scatter factor. These are produced from fibroblast. Remember fibroblast here. They will do the same function, proliferation of hepatocytes, but they enhance. Proliferation of hepatocytes is started by transforming growth factor alpha, but it is enhanced by hepatocyte growth factor. Next is vascular endothelial growth factor. They come from mesenchymal cells. Obviously, they stimulate proliferation of endothelial cells. When it is vascular, it proliferates endothelial cells and also increase vascular permeability. Fifth one is platelet-derived growth factor. The obvious source is from the platelets, macrophages in common, remember endothelial cells and smooth muscle cells. Platelet-derived growth factors, they are required for stopping the bleeding to maintain hemostasis. So it can also be associated with inflammation. So you can remember like hemotactic for neutrophil, macrophage, fibroblast, and smooth muscle cells. Sixth one is fibroblast growth factor, FGFs, commonly macrophages, produced from macrophages. This is also again, there are three chemotactics in the table, these three. This is also chemotactic and mitogenic for fibroblast. Remember fibroblast. Seventh one is the transforming growth factor beta, TGF-β. It is also commonly produced from platelet. Macrophages is in common. See, same like platelet derived. This is also, it has platelets, endothelial cells, smooth muscle cells. Platelet derived PDGF and TGF-β, they have common. Platelets, endothelial cells, and smooth muscle cells. Macrophages in common. This is also chemotactic. Three chemotactics in the table: PDGF, FGF, TGF-β. Chemotactic for leukocyte and fibroblast. Eighth one is the keratinocyte growth factor, KGF. KGF is nothing but FGF7. That is fibroblast growth factor 7 is KGF. Obvious sources from fibroblast. Since it is a keratinocyte growth factor, it stimulate keratinocyte migration, proliferation, and differentiation. Coming to individual growth factors, firstly, epidermal growth factor, EGF, and transforming growth factor alpha. These we are studying together because they belong to the same family, that is EGF family. So, they share same receptors and they have same biological activities. That is, these two factors, they bind to the same cell surface receptors. They have the same cell surface receptor. So they have same functionality as well. And the EGF receptor family. The receptor family, it is composed of four membrane receptors. They have intrinsic tyrosine kinase activity. That means once a ligand, that is a growth factor, comes and binds to the cell surface receptor, the receptor, it gets autophosphorylated because of this intrinsic tyrosine kinase. That means the receptor itself has this tyrosine kinase that gets autophosphorylated and it gets activated on its own, and the signal is transmitted to the nucleus. That is intrinsic tyrosine kinase activity, means they get autophosphorylated. So these receptors are the EGFR1 or ERBB1. ERBB1. The mutation or amplification of this receptor cause cancers of this thorax region, head and thorax, you remember, lungs, breast, coming to these belong to thorax, head and neck and brain. EGFR1 or ERBB1 mutation or amplification cause cancers of head and neck, brain, breast, lungs. EGFR2, also called ERBB2, also called HER2/neu. HER2/neu means overexpressed in breast carcinoma. Simple. Second factor is hepatocyte growth factor, HGF or scatter factor. The receptor for HGF is MET. It is MET. The MET receptor mutation or overexpression, it causes renal cancer, thyroid papillary cancer. Hence, the MET inhibitors are being evaluated for the cancer therapy. Thirdly, platelet-derived growth factors. There are three isoforms, PDGF, AA, AB, BB. These are directly active, whereas the CC and DD forms, they are in inactive state. They need to be activated by proteolytic cleavage. These PDGF proteins, these PDGF factors, they are stored in cytoplasmic granules and they have to be released by these activated platelets. There are two receptors, alpha, beta, with intrinsic tyrosine kinase activity. Coming to this vascular endothelial growth factor, which is a large family, this family consists of vascular endothelial growth factor A, B, C, D, and placental growth factor. This A wala, VEGF-A, is the major angiogenic factor. Major angiogenic factor. It is the major angiogenic factor. B and placental. Placental means having to do with fetus embryo. Placental. B, baby. Baby and placenta. VEGF-B and placental growth factor, embryonic vessel development. Whereas C and D, again, they are also angiogenesis. Apart from angiogenesis, lymphangiogenesis as well, lymph vessel development. This VEGF, they are required for maintenance of normal endothelium. To maintain normal endothelium, if VEGF is not present, the normal endothelium is lost. It can create a ruckus like by increasing vascular permeability. This VEGF, they are highly expressed in epithelial cells adjacent to fenestrated epithelium. For example, in kidney podocytes and retinal pigment epithelium. They affect vascular dilatation and also increase vascular permeability. These are the functions of VEGF. These receptors, VEGF receptors are 3123. VEGFR1, VEGFR2, VEGFR3. Antibodies against VEGF factor, they are being used as a treatment of tumors, for treatment of tumors like renal and colon cancers. These renal and colon cancers, in order to grow and spread, they require angiogenesis. So when you stop the angiogenesis, when you suppress the angiogenesis, the growth and spread of these renal colon cancers can be hold back, can be withhold. So the antibodies against VEGF are being used in the treatment of renal and colon cancers. Anti-VEGF therapies are being used in wet age-related macular degeneration, retinopathy of prematurity, diabetic macular edema. In pregnancy, there is increased levels of soluble VEGFR1. When these soluble vascular endothelial growth factor receptor 1 increases, they cause sequestration of free VEGF. They destroy the free form of vascular endothelial growth factor. When the vascular endothelial growth factor is not there, the normal endothelium is not maintained. So, because of increased vascular permeability, there will be proteinuria and hypertension. That leads to preeclampsia of pregnancy. Now coming to fibroblast growth factor, there are two forms, acidic and basic. Acidic FGF, AFGF or FGF1. Basic is BFGF, FGF2. The fibroblast growth factor 7, as we have discussed in the table, FGF7 is also called KGF, keratinocyte growth factor. This extracellular matrix, it acts as a reservoir for FGF. As soon as the FGFs are released in the bloodstream, they get associated. As soon as they are released, they are associated. They associate with heparan sulfate present in the extracellular matrix and is stored there. And they are released whenever required by proteolysis. This FGF signal through, even these have this 4 tyrosine kinase receptors. They promote wound healing. These FGFs promote wound healing. Transforming growth factor beta, TGF beta, it has three isoforms, TGF beta 1, beta 2, beta 3. This family also includes bone morphogenetic proteins, activins, inhibins, Mullerian inhibiting substances. This TGF-beta has a role in scar formation and also in fibrosis of lungs, liver, intestines, and kidney in conditions of chronic inflammation. Whenever there's chronic inflammation of these organs, fibrosis is seen because of TGF-beta. Yes. Thank you, everyone, and all of you have a nice day.